You're watching CTOM TV. Knowledge is power.
Pillar one, genetic testing. So genes can be altered in three different ways to cause cancer. You can get what we call fusion genes, where two genes are spliced together. And so a fusion gene basically is a mechanism where one component of the gene gets spliced onto another component of another gene. And typically what happens is in cancer is you will get a gene that's supposed to be turned off, like a gene that's only active in embryonic activity, and then gets turned off for the rest of your life, gets fused to a gene that's turned on 24/7. So in other words, you'll have this gene that's involved in digesting a certain protein in your body, and it's needed to stay on all the time. And what will happen is you'll get this event where the control region of that gene that tells it to stay turned on all the time gets spliced onto a gene that's supposed to be turned off, that's involved in growth and development. And you get this fusion gene where a gene that's normally turned off is now turned on all the time. And this is called a fusion gene. Okay, so the next one is a mutation. And so in this case, the mutation causes a change in the structure of the gene. So when it binds onto its receptor or its partner, in order to activate a signaling pathway, these signaling pathways are usually involved in growing and development and copying of cell structures and so on, a cancerous process. What happens is it gets stuck to it like glue. So it's like stepping on the gas pedal of a car to go up a hill and then having the linkage in your gas pedal break off while it's on full blast and having your car stay on full blast going up the hill. So that's what mutations do typically, is when they occur in oncogenes, that is. So when they occur in cancer-causing genes, they will cause that gene to stay stuck, turned on. When they occur in tumor suppressor genes, and tumor suppressor genes are a class of genes that prevent tumors from growing, basically what they do is they inactivate those tumor suppressor genes so they can't function anymore. So depending on the gene's function, the outcome will be either the gene's turned off completely or turned on all the time. So overexpression, this is something that basically refers to how much of the gene is produced. So every gene will have a certain amount that it's required in the cell. And obviously, like anything else, too much is not a good thing. So it's like, having 2 glasses of wine is a great thing. Having 2 bottles of wine is, it might feel good in the moment, but you can have a lot of side effects from that. So it's very much the same as with genes. You need a little bit at the right amount, or the right amount at the right time, and then that gene gets turned off. Overexpression occurs in genes that are typically involved with growth and development, and the cancer cells basically turn the genes on and they stay turned on. When we refer to overexpression, we're referring to overexpression without a mechanism. In other words, the fusion gene also causes, constitutive signaling, as we call it. Mutations also cause constitutive signaling, and overexpression can cause constitutive signaling. So all 3 of these are mechanisms that cause a signaling pathway to stay turned on all the time. Overexpression is just typically what we call an epigenetic. In cancer, it's an epigenetic mechanism. And it basically is the switch. It's like the light switch gets turned on and it gets stuck turned on. Typically, there's a process of acetylation and methylation in epigenetics that gets fairly complicated. But it's what we call a control region or promoter region of the gene. When it has what we call methyl tags on it, it's turned off. And when those methyl tags are removed, then the gene gets turned on and activated. So typically in epigenetic changes, what happens is something happens to the control region that turns on the gene when it's not supposed to be turned on, and it's turned on too high. So that's overexpression. So that's one of the other mechanisms, apart from fusion genes and mutations, that can happen to genes that cause them to become oncogenic.
Thank you for watching Cetome TV. Subscribe below and stay informed.